So by now, what we've done is we've gone through 4 chapters. We've made a cell. I mean, if you think about what chapter 4 is all about, you think about the fact that there are different types of cells. What we're actually doing now is going a little bit backwards, and we're looking at what is the physiology of plasma membranes. And a couple of things to think about here is one thing that I tried to, the first of the things I would like you to think about. One is, remember, not only do we have a plasma membrane, so this is the outside of some prototypic cell, and there's a nuclear membrane. Remember that each of these substructures, these organelles, if you will, okay, each of these are made up of membranes. And we're focusing primarily on the plasma membrane of the outside of a cell here. And this is you remember these are phospholipids. It's a lipid bilayer. It's an amphipathic structure. And I want you to think about something before we get started, and that is, how does stuff get in and out of a cell if you have a plasma membrane? Number one. Number two, if you have membranes surrounding this, and we can begin to think about how things move in and out, what is it inside the solutions, both inside and outside of the cell, that allow it to maintain its integrity? I mean, think about that for just a second. If you're made up of trillions of cells, and those cells have to maintain a specific shape, and you understand some of the basic mechanics of thermodynamics, you understand that, say, water is going to move down its concentration gradient. We call that osmosis. Molecules will move from an area of higher concentration to an area of lower concentration. We call that diffusion. If that's happening, how then does Mother Nature regulate who's moving in which direction and why? So that's number two. And then finally, what's the consequence of that movement? And I'm going to give you a sort of little snapshot into what this is all going to look like at the end. Obviously, yes, we have to be able to move things in and out of a cell. If you think about how it is you ingest food and poop out materials afterwards, it's the same general idea. You must move materials in, extract what you need out of them, and then remove them. A cell does exactly the same thing. But here's a major difference. Depending upon the type of cell that you're actually looking at, and remembering that cells are used to make up tissues, and remembering that I've drawn for you this idea that tissues can be muscular, they can be neuronal, they can be connective, or they can be epithelial. And you may not know what any of this means right now, and that's completely fine. But here's what I want you to think about. How is it Mother Nature, by this really simple design of a plasma membrane, can create a subset of tissues that are excitable, and then others that are used primarily as insulation? Connective tissues, epithelial tissues. They may have a resting membrane potential. They may not have a resting membrane potential that is used to generate either an electrical signaling system, as you see inside the nervous system, or an electrical signaling system that will regulate muscular activity, as you see inside smooth muscle, skeletal muscle, and cardiac muscle. So understanding how a plasma membrane is actually constructed is a lot more important than it may seem. And even though I think this chapter is constructed so you could skip right over it, if you didn't really want to understand all of this material, as a biology student, you want to understand what it was that caused the ability of, say, one cell to begin to communicate effectively inside a population, where something inside of the solution allows this to happen. And that wasn't just a pun. Okay, so now that I've told you that, plasma membranes have obviously pretty obvious, responsibilities. By defining the outer border of all cells and organelles, they manage what enters and exits a cell. I already told you that. They receive external signals and initiate cellular responses. We need to be able to talk with one another. And you also need to be able to hold on to one another, right? Literally. If you think about the fact that every single one of your cells is held together, right? And how that actually works is kind of amazing when you actually put the whole story together. And here's a fluid mosaic model, right? I've shown this to you before. You can see this phospholipid bilayer here, and there's that cholesterol. That's what's responsible for modulating, along with temperature, the fluidity, how these things actually move inside of the space. And you have these integral membrane proteins, some peripheral membrane proteins, and modifications to these. Imagine this is a glycoprotein. What it means is there's some form of a sugar. Remember those, in this case, covalently attached glycosidic bonds of some sugar attached to a protein, glycoprotein. Glycolipid, some dehydration synthesis reactions attaching sugars together, so they attach to the glycerol backbone of our lipid here. Okay, so this glycolipid has sugar on it. And these are really important. This is how you tell, literally how individual cells tell themselves apart between not only cells that are adjacent to one another, but cells that make up your body versus somebody else's body. Okay? Here's those phospholipids again. We've already been through their basic instruction, so in case somebody skipped over this inside Chapter 3, you can make sure it's you have this opportunity to review for your exam. There's that glycerol backbone. There's that charged phosphate. Here are non-polar lipid tails, fatty acids, and this one here is unsaturated, and this one here is saturated. Remember that the head group here is literally hydrophilic. The fatty acid tails are hydrophobic, and this means that you actually, when you put them tail to tail, they create that amphipathic structure. Here you can see what would happen if we just made a preparation of them. In fact, in class, I would have taught you how we would do this inside lab, but the reality is inside of a liquid substance, these literally fold to make spheres on top of themselves. And inside your cell, they literally do this, but they also do this. Okay, they tend not to make a lipid bilayer sheet because water would then act here, but the forces of water actually create the spherical shape for 360 degrees. Now, there are many different types of proteins that can be inserted. Now, remember in Chapter 3, we went through the different types of macromolecules that could actually be made, and if these are proteins, they could either be transporters, receptors, enzymes, and/or binding and signaling adhesion responsibilities. Now, you can see inside this cartoon, you have that peripheral membrane protein here. They only occur on the surfaces, internal and external. But the membrane is actually asymmetrical. You'll notice that our glycosylations are only on the outside, whereas our cytoskeletal proteins are only on the inside. So when you're looking at this, you're like, oh, okay, I can see what Mother Nature is doing. She's creating an asymmetric representation of a barrier from the outside world to the inside world. And that's what that protein channel is going to be doing. Integral membrane proteins, there are multiple ways to construct these. You're going to see that I'm going to walk you through an experimental design that we did some years back to look at mutations that drive mutagenic steps in cancer and specific types of breast cancers. All right, what you're looking at here is a family of integral membrane proteins, and the one family we're going to be talking about is the epidermal growth factor receptor family, and they have a single transmembrane spanning area of amino acids that have to pass through this. But you can have multiple types of proteins that spend a lot of energy designing the amino acids that will allow them to stay inside this non-polar region here, where the polar proteins would be on the outside. And so to tell you about this experiment would mean that in one type of breast cancer, a specific integral membrane protein expressed on a series of epithelial cells is mutated. And it's mutated, let's go back, it's mutated at the beginning points of the, in this case, what we believed was the beginning points or the ending points of the transmembrane domain. There's something going on here that we still haven't figured out, and it's been many years since people have started this line of inquiry, where what we said is, look, we want to see if we can artificially engineer that transformation step that naturally occurs. So in this epidermal growth factor receptor family member, we're going to call it ERB2, ERB2. What we did is we literally engineered in mutations inside different locations here. And now I want you to think about what we would do, okay? If you remember that amino acids are polar, non-polar, positively charged, negatively charged, or they have those large aromatic hydrocarbons that are attached to the outside. How would you go about designing now the mutations that you would find here? Well, we had a pretty smart kid inside the lab at the time, and really what he said, he's like, well, you know, it just makes sense to me to make conservative substitutions from the normally occurring amino acids that were here. And that's what, at the time, the graduate student in the lab was Christine Burke, and that's what she actually set about doing. She worked with Chris Lemon, who was up in the molecular biophysics lab where they would do the crystallography piece, and they would look inside the non-polar regions here, right? Because this is what would be sitting inside that interface between the phospholipid bilayers. So they set about making a series of substitutions. They demonstrated that a family of isoleucine, and I believe it was leucine, valine and isoleucine, could drive transformation phenotypes inside of epithelial cells. But the interesting thing is this, is you can make those mutations. You can drive the transformation, but those mutations were never found to be naturally occurring in populations with breast cancer. So even though you could experimentally design a mechanism to drive that, it would be another 5 or 8 years before people figured out exactly how this mechanism worked. Take-home message from this explanation. You can have mechanisms that you need to understand. These would have to be polar amino acids out here. These have to be polar amino acids out here. They could not be inside here. So when you set about making those mutations, it had to allow for the protein to look as though it would normally be behaving inside of a breast cancer tissue. Okay? Now, having told you that, those modifications here, they function in cell-cell recognition and attachment. They literally are used for the ability for me, if, let's imagine we were inside a class, I'm going to point at 2 random students and we say, I'm going to take the liver out of you and I'm going to put it inside that person over there. And what we're doing then is we've screened for, the last time I actually ever thought about this, at least 26 different types of antigens that could be identified, and those are the glycoproteins and the glycolipids that would say, okay, they're close enough in the similarities of the carbohydrates on the surface of their membranes so that the immune system wouldn't go and just simply destroy that tissue. What it also means, of course, is that now that patient who's received that piece of liver will now have to be on some immunosuppressant drug to sort of lower the likelihood that they're going to find something that doesn't belong here, meaning the immune system won't find something. And to see that, take a look at this. How these all work is, if you could imagine, here's an example where our own immune system, T cells, CD4 cells, are also called helper cells. These helper cells were initially identified, I should say, they were identified as initially the original receptor for HIV infections. This has to be, oh my God, at least 30 years ago now, right? 25 years ago at least. And of course that turns out not to be completely true. Work with another friend of mine, Steve DeGar, he identified the co-receptor that's required for this. And what he demonstrated was that HIV causes the activation of these receptors along with a co-receptor, and then that's what actually causes now the movement of the HIV infectious particle into the cell. Now that's not how it's supposed to work. It turns out that HIV, the virus, co-opts the normal function of this glycoprotein on the outside of the cell, and that allowed for the receptor-mediated endocytosis to occur. So Mother Nature is always looking. There's a couple things you need to figure out in this class right away. One of which is you're just simply a bag of salt water. And because you're just simply a bag of salt water, the other part to that is you're a meal to something else. And you may not understand that just yet, but what this means is whatever HIV is hoping to accomplish, it's looking at just this population of cells that have CD4 and CCRK5 and glycoprotein 41. There's a triumvirate of proteins that ultimately were discovered for this to work, right? And the way this actually happens is Mother Nature is like, well, HIV is like, I want my needs met. And the cell is like, well, you figured out how to get inside of us, and we don't know how to stop that. Well, when we do stop it, it ends up killing the patient. So this gets us to what does it mean to actually have fluidity? You know, we're talking about you actually have different types of phospholipids. We teach you this general idea of how they're made, but there are many different types of phospholipids and how they're packaged. Saturated and unsaturated will create more packing disorders. And by that I mean unsaturated fatty acids versus saturated fatty acids, which are more rigid. When things like temperature change or cholesterol concentrations change, these all combine to create problems for cells as they need to change their shapes. Normally I would describe how red blood cells now move through capillary beds, and I'll draw a capillary bed for you. All right, yeah, I can do that. Look, here's a capillary bed. And red blood cells are packed inside this tiny little tube, and they pass through this tiny little space, and ultimately they discharge out oxygen and pick up carbon dioxide. So let's put the carbon dioxide moving in this way, right? So the general idea. But the red blood cell is passing through here. Now, if that red blood cell is made by a large number of saturated fatty acids, then that red blood cell is going to be more rigid. Likewise, if it's really cold, it's going to be more rigid. And if you have really high levels of really bad types of cholesterol, then the rigidity of this will actually cause those cells to fail. Never what you want to have happen inside of a capillary bed. And I'm not even telling you it's what actually does happen. I'm trying to give you a mechanism to understand how it would happen. Okay, so now having told you that, well, like I said, the PowerPoints will be up on Blackboard. You can go watch this. It's just going to vibrate around for you and move around like if you think about how you boats floating on the water. It's pretty much the same idea. Now, transport is when things get a little bit more complicated. And really what I want you to start thinking about is by the time you've got to this point, or hopefully you've gotten to this point inside your reading, inside your textbook, right? You're starting to think to yourself, well, this actually means that we actually have a membrane that's selectively permeable. It can't be impermeable. We have to have materials moving through and forth. And one of the things I want to teach you immediately is, let's go here, and I'm just going to draw in on top of this. Maybe this will work, maybe it won't. Imagine, if you will, oxygen has to get inside here. Well, it can just pass. It's a gas. It's small enough it can do that. Likewise, carbon dioxide can pass from inside of the cell to outside of the cell. But remember, these are non-polar structures, so they won't interact with the tails here, and they won't interact with the heads there. But let's imagine it's something like water. And you remember, water is a unique molecule, and that water, guess what? Well, it doesn't want to go anywhere near here because that's non-polar, and that's polar covalent, so it's polar. So Mother Nature literally has to build a series of transporters called aquaporins. I'll put that in there for you. Porins, there are multiple types, 1 and 2, and they allow now for water to move through here. But that also means that the amino acids that Mother Nature had to put inside here, at least on the inside face of this transporter, they had to be polar. On the outside face of the transporter, they had to be non-polar. And I'm hoping you're beginning to see the significance of that. Okay, one more idea before I pause here, and that's going to be how this would actually work would be for us to think about what's the difference between having ions on the outside of the membrane and ions on the inside of the membrane. And then what happens if something actually requires energy to move it against its concentration gradient. And here's where I want to introduce this concept. This would be a low concentration of something, and this would be a high concentration of something, regardless of what it is. And when we're thinking about thermodynamics and how thermodynamics actually work, remember, diffusion materials always move from an area of high concentration to an area of low concentration. Okay? So there's no getting around that, but that's passive and that doesn't require energy. However, if we want to push something against its concentration gradient, you would require energy to do that. Okay, that's a pretty good place to stop. It's also a pretty good place where we'll pick up.